Hello, everyone. Welcome back. I am Jackson, as always, and we've got Enzo here. We also have Standing for Truth, and we're going to talk about stuff and things, very likely. So I will give the floor to him and let him talk. So go ahead.
Sorry, I heard an echo there. How we doing, guys? Do I have any creationist friends in there? Probably not, I'm guessing. In the chat area, we got. All right, perfect. Yeah, I'm standing for truth. I've seen some of your videos, Jackson. You got some interesting stuff, so I think it should be a interesting conversation.
Mm-hmm.
Did you want me to kind of just present where I'm coming from for a couple minutes, and we'll just go back and forth there?
Yeah, sure. Go ahead.
Okay. I think it all comes down to obviously the empirical evidence. We're looking at the same evidence, and what are we inferring from there, right? Like in the biblical model, for example, we do believe in change. Obviously, biological evolution means change in allele frequency and populations over time. I'm sure we can agree to that. The question is, do those changes have limits? And I'm sure you've heard that a million times, and I think we'll go in some pretty good depth into that question. Because, I mean, according to us, I think as biblical creationists, we would say that God initially, he would have put a considerable amount of information into our genomes, right? So they're stored in our genomes in compressed, hidden form. So therefore, when this information, say, is decompressed, even deciphered, revealed, or unscrambled, at the end of the day, that can't really be used as evidence for, say, large-scale pond-scum-to-people type evolution, because the changes in variation that we're going to see from those new traits, that information was already present and stored in the genome. So I mean, it goes back to limits. Do I believe that I can get a dog as small as a flea? No. Whether it's physical, whether it's genomic. Do I believe I can get a dog, say, as small as big as an elephant? Because we do know there's animals as big as an elephant, obviously, like an elephant. No. Once again, so I believe there's limits there. You might say that's physical limits based on physics and factors like that. And at the end of the day, like Adam and Eve, they would have been given a significant amount of design variation. So these, best way I could put it is these built-in alternatives God put into Adam and Eve, they'd be scrambled over time, and new traits could arise during this process. So when we get into the question, and I know you'll have a sufficient answer for this, but what is that observable mechanism, Jackson, that adds new novel information to the genome, right? Because normally what I see as answers, and I think you might have different answers to this, but, I'll see, say, sickle cell anemia. But when you actually look at that, you're seeing like a resistance to the malaria parasite, but normally that's by producing deformed hemoglobin molecules, right? So there's obviously a loss of information, a loss of function there,
Wait,
and you're not going to get rich.
I'm sorry. I think I missed something. Say, what were you saying about the parasite?
Like with sickle cell anemia, right? It confers a resistance to malaria. Okay.
Yeah, I got you.
But if you actually look at it, it's producing deformed hemoglobin molecules. So at the end of the day, the question is, how are you going to get rich by losing money? Even aerobic citrate digestion by bacteria, which we can get into. If you actually look at the literature, it involves the loss of control of the normal anaerobic citrate digestion. So once again, I mean, it's involving the decay of prior information. So as biblical creationists, we believe God wrote the genetic script, and now we're just seeing variations based on that initial genetic script, right? So those types of examples are consistent with biblical creation, but I think the big thing in our model would be genetic entropy. And I'm just talking about biological entropy, as in, say, the degeneration of our information systems based on those accumulation of mutations over time. So at the end of the day, that'd be consistent, say, with the fall. And that's what we'd be seeing, from then on. We'd be seeing decay, we'd be seeing change, but at the end of the day, that change is limited. And you can go ahead, Jackson.
Okay, so I wrote down a few things, but I think I want to start with the first of all, I do want to ask you, what do you mean when you say information? You use that word repeatedly, and so I want to ask what specifically what you mean by that.
I would say because our genomes are obviously made of very specific information. Like if I look at the English language, the English language is made out of 26 letters, 4 letters, obviously when it comes to DNA, and we can see obviously English books. Like for example, if so since you're asking me what information is, do you believe, say with misspellings and typographical errors that say that English book made up of 26 letters, made up of that information, uh, could ever turn into, say, like a German book or a Korean book? I guess I'm using the analogy of the English language, but could that happen, do you think?
Well, our language has changed significantly from High German to Old English into early English to modern English. Our language has changed significantly.
Wouldn't that be based on more of like intelligent input? Like, for example, the word
Not
computer,
necessarily.
obviously, well, obviously, like the word computer, it didn't exist 200 years ago. It exists now due to intelligent input. But would there be
Um,
any intelligent input, say, in the change we see in living organisms?
oh, I think. Well, the analogy with language is uh, the point of using analogies is to make things more digestible for an audience to understand, and so to we can use the analogy with language. I think very well. I think it works very well because there are lots of accidental things that occur in language, and you can look up the etymology of certain words and whatnot, and you can see when a word gained, for instance, an E or an A or something like that, that changed it into its modern spelling. And so we can see the accidental changes of these words over time. We can see that adding up, and we can watch as the language as a whole changes over the centuries. Um,
But I mean,
and
once
so,
again, like, isn't that based on more intelligent? Like, obviously, molecules to man type evolution. I mean, there's not really any intelligent input. It's just based on natural selection and random
well,
variation,
not
mutation.
well, not necessarily. There are examples of words where there was an accidental spelling and that became popular because it was written in a novel or something like that, and so that new spelling, which was an accidental misspelling, became the new spelling of that word. And so I wouldn't say that that was necessarily intelligent.
Wouldn't
It's
that be like alteration of, like, say, the pre-existing, in that case, English language? Like if we're making
yes,
changes and alterations of, say,
yes.
the existing genetic information. I mean, are we really adding any new novel information? Like obviously, we see examples on
Well,
Google of these information gaining mutations. What would be the best example, would you say, Jackson?
well, see, then that's where the analogy breaks down because with English, uh, I mean, you can make up new words, but ultimately all words are made up. Um, and we can choose to define words however we please and whatnot. But with genes, you can have a section of nucleotides that's totally non-functional become functional and specify a very important gene in an organism, and that has been documented in a number of examples. Like I think notothenioid ice fish, for instance, they had a section of nucleotides that didn't do anything, and it became useful, and it became the section of the genes that specify their antifreeze glycoproteins.
Was that
Uh,
based on like it? Was there a gene duplication involved in there too?
in that one, no, there are. Um, or wait, no, I think that was the codfish. I think the notothenioids were a different one. I think that was in the you have that was in the Atlantic. The codfish, the ice codfish. I think it's the notothenioids that were the ones who duplicated it repeatedly. Although you also have it in the zoarcid uh, uh, ice fish, who basically they just duplicated this gene that was called it produced SAS, I think, or it was called SAS. It basically had a minor antifreeze function that was basically amplified, although that's not what happened in the notothenoids, which was they had a gene that specified a trypsinogen, which is makes, if I remember correctly, makes trypsin, which is an enzyme. But this gene, which had a functionally unrelated, which was functionally unrelated, was duplicated over and over and over, and mutations occurred in it, and it gained this new function, which was specifying the antifreeze glycoproteins, and so that allows these notothenoid fish to live in the Antarctic
But like,
waters. So that's just one example, because genetic duplications are often a very good basis for generating new structures and functions. The example I like to give with the Ragavelia water strider. Ragavelia is a genus of water strider. They basically had duplications in two genes. They're Geisha and mother of Geisha. And so when the duplications happened, mutations occurred in these, and it caused them to develop these new propeller fans, which with these new propeller fans that didn't previously exist, ergo they didn't already have the mutations in them to generate these because they didn't have the duplicated form already. These new structures appeared and allowed them to invade a new niche, which means they can go upstream now instead of just going downstream, which is what all the other water striders can do. So there are a very large number of documented cases of new structures, functions, things like that arising. It's not simply about heterozygotic advantage like in the case of sickle cell anemia, although that is a popular example. But it's not the example I would give when talking about beneficial mutations.
but are these changes actually adding the novel information? And like, is it something meaningful, or is it like, because you're saying it's a gene that was useless, but now
Not
based on, say, a
necessarily.
mutation, or it wasn't functioning, so it wasn't
No,
functioning, but
not
based on, say, this mutation, it's now begun to function, and a new trait or a new feature
not
has arisen, right?
necessarily. No. In the case of Ragavelia, the water strider, the genes that were duplicated, Geisha and mother of Geisha, were employed in some other function. I don't remember what, but they did have their own function. But the duplicated forms of these contributed to making this new structure, these new propeller fans that they didn't previously have. And so this is a new structure that was developed because mutations occurred in these certain duplicated forms of these genes, which are gene duplication is itself a mutation. And
But
so
like,
mhm.
according to our model, and I get what you're saying, and I want to comment on the gene duplication as well. According to our model, like the species, they're not static. Obviously, we see that change, but so therefore neither are the genomes. So the changes that we see over time, sometimes they're randomly, but other times it could even be in pre-planned pathways, right? According to, say, instruction from a pre-existing algorithm. So what you're saying, I mean, whether that new feature, that new trait, that novelty is being now emphasized based on the pre-existing information, at the end of the day, it's not really, demonstrating a new novel or meaningful information. Like with gene duplication, maybe correct me if I'm wrong, like the effect of a gene, it often depends on a gene copy number. So if that organism, say, appears with extra copies of a certain gene, as far as I'm like, it may not be able to control that expression of that gene, and therefore an imbalance is going to occur in its physiology. Overall, it's going to decrease the fitness. And I think Down syndrome is actually is an abnormality due to that.
Yes, it's trisomy 21, I think, or
Yeah.
23, one of those.
So obviously, that's changing the information. It's still, it's obviously still deleterious. And like, if I gave you a copy of a book you already have, and then, there's not really any new information. If I started, say, putting misspellings or typographical errors in that
So
second or third copy of the book, don't you just have, a more messed up, degenerated copy of, say, that original book? Like, is
so
that going to build a genome, Jackson?
let's cut through the analogies. Analogies only get us so far. Let's actually look at the data. The data we have here is that we have two duplicated copies of genes, and there are mutations that occurred in these genes that gave an organism new structures and allowed it to colonize a new environment. That's not an analogy. That actually happened. We can measure that happened. And so we are talking about a new structure that formed due to mutations. And this is just one of a large,
But
long list of examples. And I've been doing a lot of research on them recently because of RJ and I are we're critiquing the Answers in Genesis books, and so I've had to go through a lot of the sections
What's
on
the new
genetic
feature exactly again? Just
propeller fans. I think the paper is like it's like taxon. There's like novel structure, taxon restricted something. Oh crap! I can look it up. Don't have it at my fingertips, but I can look it up at the moment because it's one of the papers I like to cite generally. Taxon restricted genes at the origin of a novel trait, allowing access to a new environment.
what's like,
I'll try
and
to
what's
just
the exact? What's the exact? Like, is it a deletion? Are we
no.
seeing like a gene deletion as well
No,
involved in this?
it's not a deletion. It's a duplication of two genes, Geisha and Mother Geisha, and you have mutations that are occurring in these, which are which caused it to develop this new structure, which is the propeller fans. And so what you've got a new structure that occurred because of mutations. That's not an analogy. That actually happened. And so it allowed it to colonize a new environment. And so you do. I mean, I think you have to recognize that that's what happened. And this isn't the only example.
But I'm saying like obviously natural selection is, a selection process. When you get this duplicated gene and natural selection is now acting upon this duplicated gene and mutations are, acting upon that duplicated gene as well. And then you're getting these new features and these new traits, is that really acting on meaningful information is what I'm saying, or is it just
I
acting
mean,
on pre-existing information?
well, wait a minute. So are you drawing a distinction between meaningful and pre-existing? If so,
Yeah,
what
like,
is your distinction?
was this change or alteration possible based on the original genetic script, whether it's duplicating the original information, whether it's changing it by any means, unscrambling, deciphering? I mean, was this change based on that original genetic algorithm is my question, or is it just something that is a
Well,
molecules to man type evolutionary change?
all mutations are on pre-existing DNA. You can't have a mutation that didn't occur in pre-existing DNA. I don't know how that's kind of a paradoxical thing to think
Well, then
about.
how are you going to build the genome? Like, obviously, to me, God wrote the genetic script. To you,
So
you're saying that evolution, natural selection, this type of evolution is a genetic script writer. So I
because
don't
I'm
understand how that script is going to go, because obviously, if we look at the analogy farm, we're looking at what? Single-celled organism, fish, amphibian, reptile, mammal. Eventually, obviously, that's going to take increases of novel amounts of information. But these types of changes you're showing me, like, I don't see
but
how that's going to do it.
again, it comes down. What do you mean when we're looking? If I were to look at the DNA with you, we would just sit down and we look at DNA. Can you point out information to me in it? Can you say this specific part is information? Would you consider a protein coding gene an information?
I would say that's I guess the best way I could put it is to say, to get from bacteria to man, whether indirectly or directly, obviously there's going to have to be a mechanism that adds genetic information, such as, and this is what I'd be looking for, is genes to obviously make novelties like arms, legs, brains. So like the tweaking of the genome, say in a bacterium, whether through duplication and mutation, it's not going to result in a human genome. But I mean, I
That's an
understand
assertion.
I think it must be assumed in your case that duplications and mutations lead to the new genes. But
We can demonstrate it. My example I just gave you is a demonstration of that fact. That this is a new structure that did not previously exist, which occurred. It was formed due to mutations, which allowed them to colonize a novel environment. It did not exist. It came into existence through mutations.
I'm
That's
saying
what
it
this
came into
is.
existence through that original genetic script, because technically the burden of proof would lie, say, with you,
It lies
Jackson,
with
who
both
are
of us
saying that duplications and mutations can, say, lead from fish to fisherman, but not with those that don't, because we don't believe that new novel information, because obviously we're seeing more so degradation, we're seeing degeneration. Like, are you familiar obviously with like neutral mutations?
Am I familiar with them? Yes. The vast majority of mutations are neutral. They don't do anything. Although they can. They can come to have function, as in the case of the codfish. It had neutral mutations in this section of DNA, but eventually it came to express this new function. Same with bacteria. You can replace their promoter sequences with like 100 base pair sequences, and within like a few mutations, it becomes a functional promoter sequence.
But aren't we seeing, say, like with the ENCODE project, that most nucleotides, they're playing now a role, say, in multiple overlapping codes. So that means the beneficial mutations that we were kind of talking about earlier, which are not, say, deleterious at some level, aren't they vanishingly rare? Like, how often do we see it? Because obviously the majority of mutations, like you said, are neutral, and I agree with that. Are the beneficial mutations, the ones that would be amplified, say, by natural selection, are those more so rare?
Are they rare? Yes. Both harmful and beneficial mutations are more rare than neutral mutations, because the code is, because a large portion of our code doesn't do much of anything, and because even the amino acids that are important to us are called redundant or degenerate, which sounds very malicious in my opinion, but that's one of the words that describe them.
But the thing is, we're seeing from like...
Hold on. Well, okay. I want to explain why this is important. Mutations are often neutral, not just because of this large portion that doesn't do anything. They're also neutral. Mutations are neutral because of a redundant amino acids code, because you have a whole bunch of different sequences that specify the exact same amino acid. Leucine, I think, is specified by 6 different sequences. And so if you were to change one of them, one of the nucleotides in the codon specifying leucine, then you might just change it to another form of leucine, or to another codon for leucine, in which case you still got leucine. Nothing changed. And so that's why the vast majority of them are neutral.
But the thing is, we're seeing now that a lot of these, say, redundant genes or regions, these, I guess, junk DNA, which would constitute a bunch of different things, we're actually seeing that they play important regulatory roles. We see that they're playing roles in even embryological development, gene expression. So we're actually seeing that the vast majority of our genome is functional to some extent, which would indicate that these neutral mutations are actually more near neutral, which means they are deleterious. We're losing information. So the best way I could put it, the bottom line is that selection, it's going to remove only, say, the worst deleterious mutations and then amplify only the best beneficial mutations. So the best way I could put it is, this means that the accumulating damage is largely invisible, because natural selection, it's not really going to act on these near neutral mutations. They're unselectable, kind of like rust on a car, while adaptations tend to be highly visible, like antibiotic resistance. So even if you, Jackson, say, present me with, I don't know, even 1,000 examples of, say, adaptation via beneficial point mutation, whatever it is, you're still failing to address the key issue, which would technically be net gain versus net loss. Because adaptation, obviously, it explains the fine tuning of the environment, because that's what we're looking at, is fine tuning. But it doesn't really explain the astounding internal workings of life in general. Okay. It does not begin to explain the mystery of the genome.
Okay. Well, you've covered an incredible range of topics.
I think it just comes down to, is our genome degenerating, right?
Okay, it's not. Genetic entropy is bogus. No geneticists take it seriously except.
Yeah, but I just demonstrated that it actually is, because these near neutral mutations, they're not selected against or for.
Okay, what Sanford does is Sanford ignores beneficial mutations. They're not part of his model, because
you just admitted they're rare.
No, I said that they're less common. I mean, in a sense, they're rare. It depends on depends on what you mean as beneficial. But okay, first, for one thing, you're bringing up a lot of topics and bringing them up in sort of ways that, for instance, let's go back to ENCODE for a moment there, because you brought that up. Do you know what ENCODE meant by functionality? When they said something, a section of DNA was functional, do you know what they meant by that?
Well, I know that they're playing more important roles, say as
No,
regulatory roles, gene
that's not what
expression
it
roles. These are still roles in general. I know
no.
what you're going to get at. You can explain it, but go ahead.
So when they said something was functional, they meant it was transcripted, which is
Sure.
indeed a large part of our gene, or a large part of our DNA is transcripted.
Yeah.
But that doesn't that doesn't parts that don't do anything are transcripted. That's that
I
it's
just told you they do stuff. They play important regulatory roles. We
no
see that
no
they're playing
no
roles
no no no
in
no
the
parts
embryo. This is function,
no
Jackson.
no not actually a lot of the parts of our DNA don't have any function. That's why the pseudogenes, for instance, don't have function. ALUs ALU retrotransposons don't have function. There are lots of retrotranspositional parts of our genome that don't have any function.
These
But
leftovers that you're talking about, they do have very important functions,
actually, a lot of them don't.
Jackson.
But that's not the point. The point I'm trying to get is, and I can demonstrate a lot of them don't have function.
Well,
Because
let me.
for one thing, actually, well, I want to finish first because this encode thing is really important. Uh, they defined functionality in such a way that it's virtually useless. Is it technically true? Sure, you can define functionality as it's transcripted and then say a large part is functional. That's not technically wrong. It's true. A large part is transcripted. But so what? That doesn't tell us literally anything about about differentiating the parts that do work from the parts that don't work. For instance, we have the gulo pseudogene, which allows us, or which doesn't allow us to synthesize ascorbic acid. I mean,
Yeah,
the
I'm
part for
familiar
that
with that.
is still transcripted, but um, it's still functional according to encode, but it doesn't do anything. It's literally broken. It has no function, and yet it has no function in the sense that it's not doing anything, and yet they call it functional. Would you consider that useful?
Well, let's just say, because according to my model,
Yes or no? Would you
everything.
consider that useful?
The thing is, we only speak 1 to 2% of the actual, DNA
Yes or no?
language.
Would you consider that useful?
It could have been at one point, and now we're experiencing the degeneration. Let me just say,
When Encode did their project where they declared that such segments as gulo were functional, would you consider that useful?
it depends what the use is, because like I said, we
It has
speak
no
1 to 2
usage.
percent of the
It's
DNA
very
code.
well studied and very well understood. It has no function.
Okay,
So yes or no, is it useful to describe it as functional?
for argument's sake, let's say that pseudogene, that specific one, doesn't have any
It
function. Let's say
doesn't.
it, but all I'm saying is, my Honda Civic, when it came off the assembly line, okay, it
Okay.
worked great. Air conditioning was working, brakes were perfect, no rust. Over time, the car has degenerated. The air conditioner broke. That just shows evidence of obviously degeneration from the fall that we have experienced these accumulations. So if something's broken, it doesn't really provide evidence for that type of evolution that you're
I'm
talking
not
about.
talking about evolution. I'm talking about is it useful to say a thing that has no function is functional? Is that a useful construct?
I'm saying that I'm going to
Yes
expect
or
some,
no?
if there's no actual, say, regulatory role, some type of role in the
It doesn't
embryo,
make any
some type of gene
any
expression, then no,
proteins. It
I would
doesn't
say it's useless.
regulate any other genes. It does nothing.
I'm
So
not saying that our genome is 100% functional, but I'm saying enough of it is functional where those neutral mutations are now near neutral mutations. They're building up in the population. We are degenerating. I mean,
so
are you denying that?
am I denying what? That neutral mutations are occurring in our population? Absolutely not.
But population geneticists themselves even agree that we are degenerating. I mean, we are accumulating
Which
these
population
mutations.
geneticist? John Sanford?
John Sanford references many population geneticists that are
Yes, he
actually
does.
evolutionists
He
in his
does,
book. They're not
like
even
Moto
creationists.
Kimura, but he
Yeah.
misunderstands and misrepresents their models to make his argument. For instance, he ignores beneficial mutations.
He already
Like I
admitted
pointed
they're
out.
rare.
They're
So,
not just rare, they occur.
but the
The
thing is,
example
natural
with
selection will amplify those. But those millions of
okay,
deleterious near neutral mutations, they're building up. You can't select those out.
that's
It's
not no no no
all
no.
possible ways.
What the point is with the with beneficial mutations is these things are. I will ask him, RJ. Um, the beneficial mutations outweigh a lot of other mutations, and that's part of what researchers have discovered. Have you actually gone through the sources that Sanford puts forth in his arguments and read what these other population geneticists like Moto Kimura have said?
I've read what they said in. I know there's a rebuttal to John Sanford's book. I think the guy's name was, I don't know, Scott or something, some type of blog. So he had some references, but no, I've never gone and actually like read their papers or anything.
I would really recommend it. Go to Google Scholar, actually read the papers that he is referencing, and go see for yourself if what he's saying is true. I always go to Google Scholar when I see something that someone has written about a paper. Like even when I get because I follow Science Magazine, even when they send me like their secondary sources, like somebody wrote an article about a technical paper, I don't want to read that article they wrote. I want to go and read the technical paper so I can see if they're accurately presenting it. You see what I'm saying?
No, I understand that, right. But I mean, and break it down for me, because obviously, you know your stuff here. This is what your education is. Well, but okay. if I'm making it simple, like, and just show me where I'm wrong here. I just kind of want to make an analogy. So okay, go ahead. I'll go after you're done.
The major problem with Sanford's argument, and I'm not saying beneficial mutations aren't rare. They, in a lot of cases, they are. I mean, we're a lot less likely to hear about beneficial mutations occurring in human populations, even though they do happen, but they
I just want to correct
very often.
I just want to correct one thing. I'll let you continue. When I say beneficial mutations, I more so mean like a beneficial outcome, because a lot of times these beneficial mutations are due to a loss of function and a loss of information. So you're still not getting rich by losing money.
I mean, although in a lot of other cases, they're caused by, for instance, gene duplications where you're adding segments and whatnot. For instance, the apolipoprotein A1 mutation in a family in Italy. Have you heard of that one?
No. You want to explain it?
Okay, so for anyone who doesn't know, the mutation, we all have a gene that specifies this protein called apolipoprotein A1. Basically, what it does is it allows us to digest cholesterol. So there's a family in Italy, and they had a mutation in their apolipoprotein A1 gene. It switched out an arginine for a cysteine. Basically, it was just this one little point mutation, and it allows they are now more likely, they're better able to digest cholesterol, to digest larger amounts of cholesterol than people are generally, which means they are less likely to get heart disease, for instance.
But it still sounds like I don't know that exact one, but it still sounds like if I look at the aerobic citrate digestion, that involved the loss of the control of the normal anaerobic citrate digestion. But what you're saying, it just seems like if I looked in the literature, and I mean, if it's a loss of control of something, if it's a gene being turned on and off, it's not really novel.
They didn't lose control of the gene, though. The gene's function was amplified.
Exactly. So it's amplified and emphasized based on, say, whether it's decompressing or unscrambling of existing information. That's not going to get your bacteria to biologists. But that's just still consistent with my model. Go ahead.
Okay, but remember, I gave an example earlier with the notothenioid fish of a section of nucleotides that did nothing, that became a functional gene. And this happens a lot. It happens in bacteria. There's actually a really interesting paper published only a few months ago about inserting 100 base pair sequences instead of promoters in bacteria, and within like one or two mutations, they turn that sequence that is random nonsense into a promoter sequence. They're not doing it consciously, of course. They have no brain, they have no control over their own genetics, but mutations are occurring such that these sequences, these segments that don't do anything, have become functional. And that's really not all that uncommon. I mean, sure, it's rare, but it does happen. The notothenioid fish is just one example. It's a really interesting example. But we do see examples of new structures, new functions, sequences that had no function that become functional. We do
Are
see
these
these
the
things
types of
actually.
changes that are going to build the genome, though, that you're
What do you
telling
mean
me
by
about?
build the genome?
Well, obviously, we're going to have to explain, this information system chock full of even meta information, overlapping codes. I mean, if we look at the phenotype, are these changes producing any type of significant phenotypic change, or is it just
Sometimes. For example, in I think Midas cichlids, they have a lot of phenotypic plasticity in their jaw shape, which has contributed to how they speciate. Um, and so, yeah, in some cases, yeah.
well?
In another case, the lizards, the Italian wall lizards on Podmakaru are another example where the scientists took a very small population of these lizards from this nearby island Podkopiste. And it's like 5 males and 5 females, and they dropped them off on Podmakaru in the 1970s. And in just over 30 years or so, these guys developed novel structures, and they directionally they
Okay,
they underwent directional selection towards becoming herbivores, and they even developed a
yeah.
cecal valve, which is what herbivores use to digest their plant material. Which, because the original population was insectivores, so they underwent directional selection and developed new structures to accommodate that.
Well, on that one, like, if I could just comment on the lizard one, because I think that's a common one too. Like that, let's say new or novel, let's say muscular valve that they found between, I think it was the small and large intestine. If you actually look at that, it's really just an enlargement of muscles already present
Yes,
in
it's an
its gut
amplification.
wall. So, in other words, far from being, a truly new novel feature, like you're saying, the shift in available food, Jackson, allowed the lizards with larger muscles at this juncture I'm referring to be more successful at feeding and reproducing. But the
So,
important thing is, let me just finish.
okay,
If
go ahead.
these lizards were returned to their original habitat, that cecal valve feature, which is cool, it's a cool variation based on, it's not proving any type of pond-scum-to-people evolution, but that may actually dwindle based on the lizard's return to an
We don't know
insectivore
that
diet, like
though.
you just said. So, I mean, that's not really,
We
a
don't know that
novel
though.
change.
That's that's an that's an assumption, but we don't have any evidence to indicate that that would actually be the case. But again, I'm left to ask, what would you expect as a as an example of something that in your mind would allow pond-scum-to-people evolution? Because I've given you examples of new structures that have arisen through mutations. I've given you examples of non-functional sequences that have become functional. So, what sort of mutation are you expecting?
Well, let me. I'll give you an answer to that. I just want to, because we kind of went on. Well, I mean, we've been on a little bit of a few rabbit trails. I just want to say one last thing about the genetic entropy, because when it comes to the near neutral mutations and when it comes to the beneficial mutations, like you're saying that he ignored, uh, you know, obviously a species, Jackson, it can undergo the minor adaptive fine tuning to its environment, like you're saying. I mean,
Mm-hmm.
that's observable. I agree, but at the same time, okay, the degeneration process is going on in many other ways. Like, to make it simple for the audience, because some of this I'm sure would go over people's head. Like, picture a 10-year-old car. It is degenerating in all possible ways, Jackson. Whether I install new windshield wipers, the question is, has the car stopped degenerating? I'm going to ask you that question. Has the car stopped degenerating if I change the brakes or switch the tires out?
You're going back to analogies. We need to look at the actual data. Analogies are fine up to a point. I don't really like to use analogies when I'm talking to people, when I'm explaining to people
I think it's a
scientific
good
concepts, but
analogy
I
for
think
what
it
we're talking
confuses the
about, though.
issue. I really honestly do think it confuses the issues. I mean, like when we talk about language evolving, changing, whatever, um, it's yeah, sure, it's sort of useful in a sort of way to explain that language does change, but language doesn't have DNA. Language doesn't have a phenotype or anything like this. It's just a construct that we've invented to help explain this process. When we're actually talking about mutations, it's better to provide a specific example of a mutation to show, hey, actually, this thing does happen. It's not just something that theoretically could happen. It actually has. We have evidence to indicate the evidence that is positively indicative of this thing actually having happened.
Well, it's because it's because in my, I'll give you the answer, because in my model, like I said at the beginning, this information, say, stored in the genome, whether it's in compressed hidden form, when it's decompressed or deciphered, these new traits it's expected, okay, because it's acting on that original genetic script. So if you want to say that, if you want to believe this bacteria type to biologist type evolution, the burden of proof is actually on you to demonstrate sufficiently that these changes based on mutation and natural selection are creating novelties. I want to see new genes that are going to be capable of creating whether it's legs, arms, say dinosaur to bird. Where's that scale information? How's that feather information going to come from that scale? Because that information wasn't present originally.
So are you aware, for instance, that feathers and hair are ultimately built from the same sort of proteins, right? They're both beta keratins or alpha beta or alpha keratins, whatever they are.
Yeah, I was just
Okay, so we have these two structures that are both derived from a singular source, which is already in the skin. So it's really just a modification, an amplification of something that already existed.
But even by you saying amplification or emphasizing a trait, you're demonstrating that these changes are coming from a pre-existing source. I mean,
exactly. All genetic
that's
changes
consistent with my model.
They have to, and that's something I asked you earlier, is how you can't possibly have a change that didn't already exist. That's a nonsensical.
If God wrote that original genetic script and now we're just seeing a variation based on that genetic script, the burden of proof, Jackson, is on you. Let
me explain. Well, first of all, the burden of proof is on both of us. If we have models at all, then we both have burden of proof to show that our models are actually true and testable, verifiable, etc. And actually, evolution is. And I can give you plenty of examples of that. But at the moment, I want to stick with the feather topic. Yes, feathers are an amplification and modification on a structure that already existed. These beta keratins, which are already in the skin. So these, through whether it's genetic drift or natural selection, sexual selection, whatever, these things became more numerous. They, grew new structures on themselves. Of course, not consciously, but they became modified slowly over time. And we can see all the structures that are in birds were actually developing long before birds were ever around. There's a really interesting paper. I think by the lady's name is Andrea Now, N-A-U. For anyone who wants to look it up, it's like the 160 million year making of the bird. It's a technical paper on traits that are in birds that were around long before birds were. For instance, the avian respiratory system, the system of air sacs within it that make it lightweight. Dinosaurs had those, or have those. Even if you don't think birds are related to dinosaurs still have the post-cranial pneumaticity. They still have the same sort of respiration system, even if you think they're entirely unrelated. The posture that theropods specifically have, which is the standing horizontally instead of vertically, like the old cartoons used to show dinosaurs, which birds also have. So that's something else that birds inherited from dinosaurs. Feathers, birds also inherited feathers from dinosaurs, because we now know that most theropods, the vast majority of theropods had feathers. In fact, we found feathers in other lineages of dinosaurs, like Psittacosaurids had feathers. Culindrodromeus, which was another ornithischian, had feathers. And so these are features that are all getting tinkered with very slightly, being independently modified. Starting over here with something that's definitely not a bird, and you're modifying and amplifying certain features until you actually get to birds. But at no point would you ever just be able to look at it and say that's the first bird.
And it's a cute just so story, but like
obviously it's not, though, because it allowed us to make predictions. It allowed us to make the prediction of Microraptor, which was verified in 2003.
But I'm saying,
Well,
wouldn't you agree that there's still a major morphological difference that exists between feathers and scales? I mean, there is still a huge difference and a huge gap
is there? I
between
mean,
such a structure.
the actually, well, really, the entirety of the evolution of the feather has already been, if I remember correct, there are a number of papers that discuss the development of feathers. They're really, really, really intricate papers that discuss this whole thing. And so, again, I encourage you to look them up. Look up the literature on the development of feathers. It's really
Well,
interesting.
if we take something like Archaeopteryx, weren't there, and correct me, weren't there fully functional feathers that existed prior to the existence of some of these apparent transitional
Yes,
creatures?
yes. In Archaeopteryx, yes, is the one of the earliest birds that we found in the record, and it
Yeah.
has feathers, which indicates that feathers were already on theropod dinosaurs before before birds branched off. And we know pterosaurs actually had these things called pycnofibers or pycnofibers, whichever one it is, which are these little thin, fine, fibrous sort of materials that are a lot like feathers, but they're not feathers.
Well,
And again,
actually, from what I understand, the earliest, say, birds discovered, you'd probably know the names of them better than me, but they actually have the fully modern features, and they're already modern in form and microscopic in detail. So, I mean, is there a big line of transitionary
yes,
evidence showing this?
yes, actually, the paper, yes, and thank you, Ration, for sending me the paper. Um, yeah, there are. You've got the. Let me see if I can find it. If it's in. Yes, you have feathered theropods because birds are highly derived coelurosaurs. They're a specific group of coelurosaurs called avialae, and so you've got these avialians who are members of a group called Maniraptora, and we already know most, if not all, Maniraptorans had feathers. So, which means that the common ancestor of birds and these other Maniraptorans already had feathers. And since we've seen that ornithischians also had feathers, either they independently developed feathers or the common ancestor had feathers. It's like which is more likely because then when you look outside the dinosauromorphs and you find the pterosaurs, they have little pycnofibers, which are a lot like feathers. And so you find this very, very slow progression of feathers. And again, I encourage you, don't take my word for anything. Go look up the literature. Don't just listen to me and take my word for it. Don't do that. No one in the chat do that. Don't ever just take my word for it. Go look up these things because if I get them wrong, then and you just take my word for it, then you'll get them wrong, and that's not good.
It depends, yeah. And when I look at these papers, like whether they're
But have you
adequate,
looked at the paper?
awesome. If they're inadequate and the evidence for such evolutionary origin is non-existent, I mean, I'll go look into that and see if it's more than just a just so
We'll
story,
see.
or
Here's why it's not a just so story. What makes the
why.
hallmark of a good model or a good theory is its ability to make predictions. Evolution makes predictions. Microraptor was one such prediction. This guy named William Beebe, back in the early 1900s, predicted that we should find, because he studied birds, and he's noticed that while birds were young, they developed feathers on their hind limbs. And so, what he or their little legs, he reasoned that there should have been a step in the evolution of birds where they had flight feathers on their front and back limbs. And we found Microraptor in 2003, which has flight feathers on its front and back limbs, a spitting image of a picture drawn 100 years earlier called Tetrapteryx. So that is a testable, verifiable model that is based on the evidence. Whoops, not my computer. So what are your thoughts on that?
Well, I mean, obviously, like I said, I need to see you know, the papers. I would have to look into them. I would have to see, because I still think that those big morphological changes are going to require that type of.
Here, look, I'll put
Uh,
one in the chat for us right now. Go ahead, keep going, keep going.
well, I mean, the best thing I can say is, with common ancestry, obviously, we're going to have to see whether or not there's limits. And what you're saying is interesting. You know, obviously, I'm going to go look at that literature with that, but I'd still like to know if you believe that there's limits. Like, do you believe we can get, say, a bacteria as big as a whale? Because we know there's animals as big as a whale, like a whale. Or do you believe there's limits there, whether it's physical, whether it's
I think the largest bacterium, or maybe it's an archaean, is a Theomargarita, if I remember correctly. But it's single-celled organisms can't get very large. There's a ratio of cell surface area to volume, and once you get beyond a certain point, it becomes more or less impossible to maintain metabolism.
Do you believe that
chemicals?
Do you believe that whales and bacteria share a common ancestor, like these single-celled organisms?
Ultimately, yes, because they share a number of similarities. I mean, of course, from bacteria, it would have to be cellular similarities, but yes.
Well, obviously, like if I looked at, say, Microsoft PowerPoint, it's going to have millions of lines of code, and Microsoft Word, the word processing program, is going to have millions of lines of code as well. And if I were to put these, Jackson, say both side by side, and let's just say I hired someone with a lot of time, energy, and maybe a government grant, they're going to find tens of thousands of identical lines of code.
I believe Kent Hovind has literally said that sentence to me.
And how is that proving universal common ancestry over, say, common design? Because obviously we know that those programs were built through common design.
It's a fair question. Again, we need to avoid using analogies because analogies obfuscate the issues. Avoid the analogy altogether because it's gonna confuse things.
Okay, so if we ignore the analogy, then
because this analogy, I want to demonstrate why this is a bad analogy. Do analogies? My tongue is my tongue is tangled. Do programs reproduce sexually or asexually?
Well, obviously, programs don't reproduce, but at the end of the day, that initial reproduction, that's a problem for you guys. I mean, how does
programs have metabolism?
No, of course not.
No, they don't. They don't have any way to transmit phenotypes from one generation to the next. They have no way to do that.
But I'm still showing you that you can look at, say, the similarities in living organisms and say the similarities in modes of transportation and conclude, wow, this is an awesome designer, whether it's of modes of transportation or whether it's modes or these living organisms. I mean,
you know what a paternity test is. You ever seen the show, like the Maury Povich show?
I don't think I've seen that show, but I am aware of, obviously, paternity tests. Okay. Yes.
So paternity tests. So in your own words, tell me what you think paternity tests do.
Well, I think they're going to show obviously similarities molecularly and see if that father is my father.
Exactly right.
Yeah.
So why does the child have the same genetics to an extent as the father? Why is that?
Well, because that genome, whether are you looking at the nuclear DNA when it comes to that?
Nuclear like obviously DNA, mitochondrial DNA. Why is this child assigned to this parent?
So you're going to be using those gene sequences. You're going to be looking at them, much like I explained how the guy's going to be looking at the lines of code, whether it's Microsoft PowerPoint or Word, and I'm going to conclude that he's my father.
Okay, but why? Why, when we look at these genes, are they concluding that this is the offspring of this father?
Well, I think we can see that. Well, I believe that we can see that through observational science here. But you're going to say that humans and, say, whales, pine trees, and elephants all share a common ancestor. So that's when you're making that giant inference based on what we can do here with operational science. I mean, is that what your ultimate conclusion would be based on these paternity tests?
So ultimately, in a sense, yes. Also, I'm really glad I laughed. I'm sorry. I'm really glad you said pine trees and elephants because I'm glad we
I
both
had to throw
watched
that
the
in
same
somewhere,
talk
right?
and we both
Obviously.
know what we're talking about. I'm very glad we both saw that.
You know
That
what? You're
makes
the
me
first
happy.
person who did know what I was talking about.
Oh, really? Oh, that's sad. I'm sorry.
Probably, I've done probably 10 discussions in the last 2 weeks, and people just didn't get it. But I mean, it
Oh,
is
that's
what it is.
unfortunate because that was a golden talk, I think. But so so the point here is we assigned this offspring to this father because of the genetic similarities. Because there's a certain suite of similarities we would expect in this child if this is the father, right? Or
Yeah.
vice versa. We test the child and we see, okay, he has this suite of genetic characteristics. Which of these males also has that one? One of them has to. And so when you look at these characteristics, when you do the same sort of paternity test, you do it on a larger scale. You've heard of like 23andMe and Ancestry.com. They do the same sorts of tests, although they do it on a larger scale.
Yeah.
They look at things like your mitochondrial haplogroups and things like that. And so what they can conclude, what they're literally in the business of doing is showing your ancestry, your human ancestry, of course, mapped across the globe.
And then you're going
Because,
to put that genomic map, next to different genomic maps of different animal groups, right? And you're going to compare the
because it's
similarities.
literally the exact same thing.
No,
You are still doing the
I
exact.
completely understand what you're saying, but like, if I were to ask you, this is a serious question.
Okay.
You know, do you have parents and grandparents?
Hmm.
Were they human?
So what you're asking is about speciation, and I'm very much happy to go into this discussion with you. So you know how speciation occurs, right? Whether
Well,
allopatric or sympatric, right? You have one species, a parent species, and it splits into daughters. Whether through the population splits and one half undergoes natural selection, whatever.
no, yeah, and I want you to explain this to me, but I just want to know, based on so obviously your parents, grandparents were human, their parents, grandparents were human. How far do you have to go back? And I understand the law of monophyly, but obviously if chimps and humans share a common ancestor, how far do you have to go back before they become non-human? What would be that time?
Well, by non-human, do you mean by Homo sapien or just homogeneous?
Well, what would be that common ancestor between chimpanzees and humans? What would be
Well,
that common ancestor?
that
Would that
well,
be like more of an ape-like
so
ancestor?
well, I mean, we both are apes by definition, but the first Homo sapiens appeared about 300,000 years ago. The first Homo species appeared, was it 2.2.5 million years ago, something like that. The ancestor between us and chimps and bonobos lived approximately um, lived approximately 5 to 7 million years ago.
But I just find it funny that all of human history, we trace that paternity test back, and our grandparents, our great grandparents, our great 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 grandparents, it's always been human. Humans have produced humans, but you're trying to say you go back long ago and far away, and all of a sudden we have these ancestors with whether it's apes, whether it's Homo erectus, whether it's, I mean, how well can you demonstrate that
Well,
empirically?
for instance, we can demonstrate absolutely that we share genetic sequences with Neanderthals. We know this because we actually have Neanderthal DNA.
Yeah, and I
And
would say that Neanderthals are human, so I have no problem with that.
so Neanderthals are a different species of human than we are. They're Homo neanderthalensis.
Well, I mean, some guy named them a different species, whether that's,
So they have,
God's classification or not. I would just say that they were a post-flood type of human that, based on adaptation, probably environmental factors, that's why you see those odd features. That's why you see those types
well,
of differences. I have no problem with that at
I
all.
mean, that goes into a whole nother slew of issues because there's absolutely zero evidence that somehow we could develop a common ancestor with another group of humans that genetically distinct from us in such a short time period. There's absolutely no evidence for that based on our life cycle and our generations and things like that.
Well,
But
based on that DNA variety, say initially after in my model, initially into post flood, because at the end of the day, if we're looking at genetic entropy now, which I understand you don't believe in,
well, no
um,
geneticists really
but
believe
if
it
if
except
there's a
Stanford.
point of least entropy, least genetic entropy, I mean those subgroups, whether it's Homo erectus, whether it's Homo neanderthalis. I mean, those were just varieties of human. That's what I would say in my model, and I don't have any problem with that based on the
Well,
DNA variety after the flood.
can I tell you something a creationist said?
Sure.
Creationist Todd Wood. Are you aware of him? He was in
He was in
Genesis.
Genesis Paradise Lost, wasn't he?
No, the other one.
Oh,
Is Genesis History? There we go. He
yeah,
was.
yeah.
Um, he says on Answers in Genesis website that Homo, that us, we are in the same kind as Homo habilis and Australopithecus sediba. So do you agree with his conclusion? He based that conclusion, mind you, on looking at the morphological similarities because we don't have DNA of habilines or Australopithecines. We just don't have it. They're too old. He looked at the morphological similarities that we share with Australopithecines and habilines and concluded based on that, and he is a creationist by all rights, believes the Earth is 10,000 years old, etc. Flood happened. He believes that we are related to these guys who have features drastically different from our own, even though we're
Well,
all still
well, the
humans.
thing is, just like there being a variation of humans based on that, those tens of thousands of DNA differences placed into each original kind, there's going to be a whole variety of ape type animals. So whether he mistaken one or the other, I mean, I would definitely classify Homo erectus, obviously humans, Homo erectus in the human category. Whether
Okay.
there's extinct type of apes, obviously Australopithecus would have just been some type of
Well,
chimp.
to him, a Sediba is a human, is in the Homo or the human holobaramid.
I'd have to go look at the A Sediba. So obviously they're looking at the bones. If I put out a
Mhm.
whole pile of dog skulls, but dogs were extinct, and you're trying to label these and categorize these, I mean, there's going to be some people making different interpretations and others making other interpretations. So I'd have to go look at the different interpretations because obviously, what's his name, Todd?
Well, Todd Wood, well, as it happens, actually, the paleoanthropologist evolutionists agree with him that A Sediba is related to us. So I think we're all on the same page in that case.
What
But
would what would
um,
ICR answer? Are you saying there's conflicting interpretations between the different
yes.
creationists?
And actually, there's a whole lot of conflicting interpretations. Some creationists classify Erectus as just an ape. Some classify Erectus, you, as a human. And it's actually RJ and I. RJ showed me a paper just the other day. RJ, can you go find it? The paper on the different people who disagree, the different creationists who disagree on the taxonomy of the humans. If you could go find that one for me, that'd be great.
I, I've seen those disagreements, but obviously, if we're looking at these fossils, these bones found in the dirt, say of
Oh,
thousands of years ago, in your case, millions of years ago, there's going to be morphological differences, and it's not always going to be easy to label
I
each in
agree.
a certain
I
category.
absolutely agree with that. I
If I
absolutely
if I showed you, and you have to agree, if I showed you, 300 different dog skulls, whether
would we
it's a
consider them
great
different species? Possibly. We might. I actually
Well,
said
we see
something
them
similar to
today, so
something
we
the
can
other
do it. But I'm saying if they were extinct thousands of years ago, you start digging them up. It's like, wow, which category? If I took all the labels off of different cars, so whether it's the Chevy Cruze or whether it's the Hyundai
Again, not a good
Elantra
example.
or a Toyota Camry, you're gonna have a
Oh, here it
difficult
is.
time categorizing. I know they don't reproduce this, that. I'm just trying to make an analogy.
Here we go. I
You're gonna
found
have
the
a tough time labeling those as whether, hey, that's from Toyota, that's from Honda. So it's understandable that they're making different interpretations based on some of the, especially if it's an extinct ape type creature that doesn't live today. You're going to have those mistakes because we're interpreting those bones
well,
based on an animal we don't see today.
oh, I wouldn't say it's a mistake. I would actually agree with Todd Wood that we are related to ACD, the but there's something I do want to ask. I don't know how much longer you want to go on.
How long have we
Um,
been going at it for?
uh, maybe about an hour by now, I think. But I do want to ask, and because you repeatedly refer to what you believe as your model, and the hallmark of a good model, because everyone has a model, but what makes a good model is the ability of said model
To be
to make,
falsifiable?
yes, A, to be falsifiable, B, to make verifiable predictions. So what would falsify your model of mutations and these variations, mutations causing just these variations within the kinds. What would
Well,
falsify that?
Well, for one, I would say that, and I'll give you that answer too, but my model really is based on decay and degeneration. And I understand we disagree about this, but I do believe that we see genetic entropy. I do see that, the universe is winding down. There is a form of, order to disorder. So I think this is consistent with the model. In regards to falsifying it, I mean, we are predicting that most of the genome is fully functional. I think we're seeing that with a lot of these genes. I think we're seeing, regulatory functions, gene expression. I would expect some type of pseudo gene, some type of junk based on that degeneration. So if we were to see that most of the genome isn't functional and it really is based on evolutionary leftovers, I think in a way that would falsify it. And I think if we've seen a ton of these information adding mutations, I do believe that that would provide sufficient evidence that these types of lower life forms could have evolved into all the life we see today. But at the end of the day, I don't see that. I just see mutations and natural selection acting upon pre-existing information. I'd have to see the opposite of that. I'd have to see
How
the opposite of that.
how does mutations and natural selection acting on genes differ from what you think I would expect as an evolutionist for evolution to occur? What do you think is the difference?
I think the difference is that, like I said before, I believe that, God wrote the initial genetic
No.
script. Now
What
we're
do you think
seeing,
is the difference for me? What do you
obviously,
think between natural selection and mutations? What is the difference between that and what you think I expect to occur in evolution?
would you agree that at one point we had lifeless chemicals that obviously evolved somehow into a single-celled organism?
Ultimately, sure, but it's not evolution. That's
No,
abiogenesis, which
I
is a
understand.
different
Yeah, no,
topic.
I understand that's abiogenesis. Obviously, you need that for the coherent theory, but at the end of the day, we're
You
talking
don't.
about change. Well,
I could honestly concede to you that God created life with the snap of his fingers, and you still have to deal with all the evolutionary data.
I understand because we're looking at change in allele frequency and population over time. So we obviously need those organisms there with the alleles, the variations of the genes to see
Right.
the change. So,
Right.
okay, so from single-celled organism to a multi-cell organism through like colonization, or how did that happen?
Yeah, actually, there's a lot of really, really interesting research with volvacines. They're a type of algae. They operate, as RJ once put it, near multicellularity. These guys are literally on the edge. I remember when I did my talk with Hovind back in the day, long, long time ago. One of the things he asked me was, "Where are the examples of two, protists going around together?" We have one, we have examples of one, and we have examples of dozens. Where's the example of the two that are together? Actually, we do have those. Gonium is an example. It's a little. I'll put it. Actually, I'll put it in the side in the open chat so everyone can see it. Gonium. It's an algae. Go look it up. It's a volvacine algae. Basically, gonium is one of these little guys, and they go around in little pairs. But interestingly, so these volvacine algae, so they can go from being from unicellularity to colonial groupings and differentiate their jobs, which obviously you would have to do if you're an organism. If you're a multicellular organism, you obviously have to differentiate your cellular jobs, but you also have to have allo recognition, where you recognize all of your cells. So you have to go through ultimately a unicellular. You have to go through this unicellular phase, and then which makes all these other cells, and you'd have to have them recognize each other and differentiate their jobs, etc. And so there's a lot of really interesting work going on in that. Again, I encourage you to look up the literature. Don't take my word for it.
Well, let's just say, for argument's sake, okay, you can demonstrate this single cell to multicellular cellular organism. Because I'm trying to. Is that what you're saying?
I mean, I wouldn't say we've worked out all this stuff. I wouldn't say that. I wouldn't go that far and be that arrogant about it. But
Well, like, is it something you can that is demonstrate? That
it
is.
is very interesting research that discusses possible mechanisms for colonializing and multicellularity. That's
Okay.
what I will say.
Well, okay. Let's say for argument's sake, now you got that, population of multicellular organisms. Would you say that those single-celled organisms? Obviously, you wouldn't say it's just one single-celled organism. Would you say there was many different ones that had? Is it like one tree of life, or is it a little bit more bushy at the bottom? Would you say?
Oh, I'd say absolutely it's bushy at the bottom
Okay.
because among prokaryotes you have horizontal gene transfer and stuff like that, and that's
Yeah, absolutely.
just genes all over the place. You got transfer of our plasmids and all this other stuff that's going on. So yeah, I'd say it's a
Okay.
bush, and then for eukaryotes, it's more of the typical tree is you see in textbooks and online and whatnot.
Now, when you actually have, I mean, when it comes to even the horizontal gene transfers, like you said, that's interesting because I would say, in a common design type model, he would give those organisms the means to say adjust to changing environments by the acquisition of pre-existing information, because that's what they're doing, right? They're transferring the different information. But when that multi-celled organism, what did that evolve into? Like, what was?
So couldn't by that reasoning you just say everything already had? We could say all of life is related. It just already had the necessary genetic algorithm, whatever you called it earlier, to create everything. Couldn't we just say that?
Well, if it's design variation, but I mean, from what I'm saying here with the single cell to multi-cell,
well, what did it evolve into?
I mean, not necessarily what it specifically evolved into, but let's say if we were to take an amoeba, okay, if we're going to make a change from an amoeba to something that has that necessary extra information, it's going to have to mutate because, and correct me if I'm wrong, like an amoeba, it has limited genetic information.
Everything has limited
information for a protoplasm.
Everything has limited amounts of genes,
Sure.
And if you're using information as genes, I would agree with that, but only in that sense.
Well, how's that amoeba going to increase, the information? Because it's going to have to need to continue to in order for, say, a heart, kidneys to develop. And if that DNA strand gets larger, say due to mutation, but just the sequence itself doesn't code for anything, right? Let's say, for example, it doesn't contain information for working lungs, heart, etc. Then the amount of DNA added, it's useless, and it would be more of a hindrance than a help. So we would have to see billions of these information gaining mutations. And if we could see that and document that, in a way that would falsify my model.
That's kind of a tall order to pull off, especially since we don't even have fossils of a lot of the protists that existed because of how small they were and things like that. What we do have, though, is, again, what I was saying earlier, is we have amplification and modification of pre-existing structures.
How did that pre-existing structures get there? That's my point.
Well, here's an example.
Here's can arise.
Well, here's an example I wanted to give. What these structures do is they arise from structures that didn't do this function originally. ATP synthase is one example. It arose from a hexamerical helicase protein that did this completely unrelated function, but was co-opted because there's loads and loads and loads of research on this topic. I did a whole video on it. It was co-opted for this function, this new function that it didn't have, that it wasn't supposed to do. And it ultimately, through a number of successive modifications, became this new structure that didn't previously exist. In one case, coenoflagellates, which I really like, and I talked about them extensively in Common Ancestry Part 5, I think. These guys have little structures in them because they're unicellular, but they can aggregate for different functions. They have structures like receptor tyrosine kinases and other parts. Like I'm drawing a blank right now, but they're these little parts that basically help cells stick together, but they're unicellular. Okay. These are things that don't need to stick together to other cells, but they're the closest relatives of the animals. And what you see when you get into the animals is these structures, cadherins and integrins. There you go. Those are the other ones. So cadherins and integrins and receptor tyrosine kinases, these things that are in coenoflagellates that are doing totally different jobs because coenoflagellates don't need to stick to any other cells. These things are being co-opted for totally different functions, which is used for cell-to-cell recognition and adhesion and signaling. These are new functions that have arisen because of mutations and natural selection.
It sounds like you tried to, not use the words this time.
I'm not.
Adaptations.
No, it is adaptation, and
Accentuated,
it probably is. It
well,
would have to be adaptation, but adaptation is a part of natural selection. Adaptation isn't. I mean, that's.
But obviously, natural selection is the selection. Well, I'll look into these different things because at the end of the day, we looked at the lizard one. Normally, the ones that people bring up, sickle cell anemia,
I didn't bring it up
digestion by bacteria. You didn't bring those up, which is good.
I didn't. Well, I didn't need to because I know what creationists think about them, and I know what's wrong with bringing up those examples, and there are better examples to bring up. With the example of sickle cell anemia, that's an example of a heterozygotic advantage. Well, there are examples of mutations in humans that don't involve heterozygotic advantage. The apolipoprotein A1 example is one of them. It's not to anyone's detriment to be a carrier for apolipoprotein A1 or to express it fully. So no detriment there. In the example of the aerobic or the aerobic respiration, the citrate, and the E. coli, the Richard Lenski research, I've done a bit of reading on that. There's some papers by Minnick that talk about how it's really not that surprising that they develop the ability to metabolize citrate because they can do it, or metabolize citrate in an anaerobic environment, I think is what it is.
Yeah. So they're losing the control of the normal anaerobic citrate digestion, and in that way, whether the genes are on or off, it's still based on that pre-existing variation.
But see, I didn't use any of those examples because I know of better examples because I've studied the data and I'm constantly reading technical literature.
And if I'm going to be honest with myself, because even the lizard one, that's another one, and obviously I use that, and I feel like I sufficiently refuted that as new. So I'll look at these.
It's a good example.
Sure, but I'll look at these certain ones that because obviously I don't know every single example that there is.
Well, I don't know every single example that there is. I mean,
obviously, I'm glad you brought up a few decent ones I can go look at and see. Hey, this looks like it is. An example of novel information, because at the end of the day, this will be the last thing I say, and you can say one last thing because we can do this again. But yeah, because according to the biblical perspective,
I like you. I'll tell you. I'll have you back on. I like you.
Yeah, I think we had fun. I think we had a couple laughs with the pine trees and elephants, but
pin elephants. I think that's on a T-shirt now. So,
and I like going up. I like having discussions with you that are going to bring up these new examples, or bring up a new. A little bit more of a challenge, because then I can go look at these afterwards. Because technically what I'm saying, with my model is the change within living things. It doesn't require the new information to be added to the genome, right? Because we expect the loss of genetic information. So I believe, obviously, whether we agree or not, I believe the overwhelming amount of mutations and changes we see, they can be categorized in, say, loss of information, loss of function. That's consistent with my model. And yes, if you can prove that, we're seeing many numerous of these, information adding mutations, and obviously natural selection adding on that, that would prove that, hey, listen, these processes, they can actually write the genetic script. Therefore, we don't need the designer to write the genetic script. All I'm saying is God wrote the genetic script. We're now seeing variation based on genetic script. So I'm going to look up these examples you said as well, if you can send them to me. That's pretty much all I have to say. If you want to,
I'll try to put them in the link or the description down at the bottom. I'll try to remember what I've said because I should probably write it down right now so I don't forget. I'll try.
Is this gonna go on right away? I'd like to probably listen to it tomorrow.
Oh, yeah. I think these things all go up automatically on YouTube. I think that's just the way to do it.
And then I'll put it on my channel, and I'll put a link to your original. I'm sure you'll be getting lots more views on yours, but yeah, I'll put it on my channel, and hopefully we can have a secondary discussion. I'll look up a few more of the examples you've said, and maybe we can kind of go back and forth on those as well. It'll be fun.
Okay. I guess to finish off, I'm still really not sure what you mean when you say information. I think there's kind of been a bit of, I don't know, maybe I wasn't comprehending what you were saying or something. Maybe it might be entirely my fault, but I have provided examples of new structures, new functions, non-coding sequences that became coding. We see, for instance, organisms adapting. No problem with that word. It's a perfectly fine word. Adapting to environments. Natural selection is shaping.
No, I agree. It's a good word.
I'm not like a pan-adaptationist. I mean, it's not it's not like everything is adapting. You know, genetic drift is most certainly a factor in things and stuff like that. But all evolution requires is changes in the
alleles.
in pre-existing genetics, the pre-existing gene.
And you keep
and
saying
all
pre-existing, right? And you just
because we have to. It's nonsensical to say a non-pre-existing.
Okay.
How would something non-pre-existing? That's nonsensical.
Everything you're saying, these examples, would you say that that's sufficient to explain the 4-dimensional genome with overlapping codes and chock full of information?
Wait,
I mean,
the what? The 4-dimensional genome?
Oh, we're seeing even one sentence of these nucleotides, they have overlapping meanings. I mean, there's so much information packed in our genome.
I'm not going to touch that at the moment because I fear that will take us down a rabbit hole. The point I'm trying to make is
that next. Okay, we can touch that next. That'll be a good topic.
The point I'm trying to make is all evolution requires is modification, amplification, whatever of structures that already exist, whether it's an arm turning into a wing, whether it's beta carotenes turning into feathers, receptor tyrosine kinases being modified to have cell-to-cell signaling. These are all perfectly well understood and explained within evolution. This is precisely what we expect to go on in evolution, and it allows us to make a model that has predictability. For instance, next time perhaps we can talk about chromosome 2, because I did a whole video on that, and it's very interesting.
Actually, I did watch that video, so I thought maybe we would touch on that. So that is a good thing. So maybe those few things we could touch on next time, because I was kind of, because obviously that's a bigger evidence. But yeah, let's touch on that one next time.
So yeah, we don't expect new structures to just pop into existence. There has to be a genetic basis for it. Everything phenotypic is based in genetics. And so what we would expect is the modification of the genes. That's it. We just expect that. And as you said, evolution is changing allele frequencies in a population over generations. The alleles, organisms are born with variation, which can be caused by, unequal crossing over, Mendelian inheritance, mutations, all these things that are coming into play to shape organisms. And that's all evolution requires. It doesn't require anything really, mysterious or weird. I mean, some things of evolution are really weird, but it doesn't ultimately, I think, fundamentally require anything weird other than organisms having variations that are inherited and that are changed through a mechanism of evolution, whether it be natural selection or genetic drift or whatever. That's really all it requires. That's the entirety of it, the sum, the start, and the finish. That's really it.
It is kind of weird that, pine trees and elephants are related because they're both eukaryotes, right?
Right. And they are. And there's it makes sense because changes are inherited.
Sure.
The first person who had apolipoprotein A1 mutation, which is a guy, it's been tracked to a guy in the early 1900s, I think. He passed that on to his family or to his progeny, who passed it on to their progeny, who passed it on to their progeny. So we can see in real time examples of a mutation or something, a variation being passed on. So we know through, that's what paternity tests are for. We know that these things are passed down and inherited. Traits are inherited, and traits can be modified. And that's really it. That's all evolution is. It's just about modification of your traits. And it allows us to make predictions, and that's why it's such a good model. That's why it's the only model currently of that is overwhelmingly accepted by researchers, is because it allows us to make predictions, and it is falsifiable, and it fits with the data.
Awesome. Jackson, thanks for having me on. You know what? Maybe next time we can touch even on some of the global flood, Noah's Ark type stuff. We'll go into chromosome 2. We'll have some fun. I had a good conversation, Jackson, and have a good night, man. I'll rewatch this tomorrow, and hopefully we'll have some more points to consider next time.
Okay. And anything that you want me to add in the description, or any more links you want me to send you, something like that, I'm happy to do that. Of course, because I'm sure I'll forget. I'm certain I will forget of something.
I'm definitely going to upload it and rewatch it tomorrow so I can get some of those examples that I haven't, because obviously if I don't know the details of them, I'm not going to be able to say, hey, was this a loss of this, loss of that, or whatever.
Yeah,
that
no, go read
I'll look at those. But I
go get on Google Scholar, look up any of this stuff.
Yeah,
That's
in the
what I
description
do.
you can just put. I think we kind of talked about models, whether it's limits or whatever. But you know what, you do what you got to do, and have a good night, man. We'll talk to you soon. Okay.
Okay, well, thank you everybody in the chat for showing up, and we are powering down. Have a good night, everybody, because it's early in the morning for me. So, good night, everybody. Bye.
Good.